All right, so what I did, I drew this plasmid. You can see on page 150. Plasmid is a really interesting way for bacteria to transfer genes between each other. They can't have sex because they do binary fission, but they can send their DNA from one bacteria to another, which is similar to what most organisms that we know about do, where you have gametes combine and then mix up genes and then come up with new variations. So very sophisticated, not sophisticated, but very simple, very effective way to switch genes, to exchange them. And we can see right here the bunch of different resistance. These are all different kinds of antibiotics. Trimethoprim. Here's resistance to disinfectants like, say, bleach for ammonia. Genes that help spread the plasmid. I'm not sure exactly what that is, but maybe they help to get them to fuse with the plasma membrane. They need to actually make this little tube to go across to another bacterium. Vancomycin, which is another antibiotic, A, B, and then penicillin. You've all heard of penicillin. I know you have. So comparing the chromosomes between prokaryotes and eukaryotes, we just have one single chromosome. Now it's circular, but it's not like you look at the bacteria and it's like a circle. It's going to be twisted up like this, just like a hose that you don't wrap up a smooth. It's going to look like that. It's not going to look like this, but it's one big circle. Eukaryotes, we have many chromosomes, many different sizes. It just depends on what species we're looking at. They're linear, so they go on one line. They don't loop around. No histones. Histones are proteins, so we'll see an image of that. This would be DNA, and it's looped around these proteins called histones. That's what these little bumps would be. Okay, kind of loop around, just keep that DNA organized. Plasma, like we just talked about, eukaryotes do not have. No organization of pairs, and we know our. And here's what I was explaining earlier. Very complex how these the DNA gets wrapped around the proteins very tightly. Of course, the only time you can really see these, oh, this is pretty cool. This electron micrograph you can see right there. Actually, this was hypothesized earlier that this is what the DNA we're doing. We knew there was DNA, we knew there was proteins, so it was thought that they were wrapped around. And to see an actual image from an electron microscope, seeing it wrapping, that's just amazing to me. And we can see as we go to mitosis, they'll start to get more compact and condensed. You actually see, and we'll see chromosomes shortly when we look at mitosis. We'll count our different onion root tip cells in different phases of mitosis. Here's a pretty cool graphic. So again, this is what we see under the microscope. When they look at bacteria replicating, they can see, okay, here's a circular chromosome. Oh, here there's a little bubble, a replication bubble forming. So this is what the interpretations. These are the two strands of DNA, and here you have the two strands copying each other. So eventually you got two new loops that are identical to each other. And so karyograms, from a karyogram, which is the actual image, we get a karyotype, which is what the karyogram, the meaning. So if we look at this one, I believe it's kind of hard for me to see. There are three chromosome 21. So there's 1, 2, 3, and that's called trisomy 21, and that's what causes Down syndrome. One of the chromosomal, changes or differences we see. So you need to be able to look at a karyogram and be able to ascertain what it's showing us. Remember, it can't show us the individual genes, but it can show us things like sex, and any kind of abnormality. So here, if you look, we see, you should see everything is paired up, only one of each, I'm sorry, pairs, you don't have any threes or ones. And then you have an XY, so that would make this right here a male. And down here, you see everything paired up, so good, good, good. And then you see two X's, so this would be female. And to those of you that don't know the symbols, we can remind ourselves, I think those were Roman, Greek symbols for Mars and Venus. Can't remember. I want to say Greek. Latin Roman. And let's move on to this karyogram. If you see one thing pop out at you, you have here, you have 3 chromosome 18s. This is called trisomy 18. This is Edwards syndrome. And what would be the gender of this individual? X and Y. You would say male, right? No, you would. And down here, we have trisomy 21. So I'll just say T21. And this is also a male. So the first gene you'll want to get is that BRCA1 or BRCA1. So you want to click on that link and follow it to Gene Cards. You can see genecards.org and BRCA1. I'll search for that and you'll see it'll pop up here, this DNA. So I'll select it, and here, BRCA1 gene. So you want the gene, codes for protein. And you look through here, and you basically want to scroll down till you get to this area right here. And this is where it wants. If you look here, you'll see it's chromosome 17. And here's the chromosome. This is called the P arm and the Q arm. So it's located on that Q arm, and it'll show you right here. Q arm, section 2, subsection 1. It's like section 2, subsection 1, and then 31. And I'll show you later what that is exactly. But you write down here, this is the length, the location actually. It's 43,044,295 to 243. And I'm sorry, 43,044,295 to 43,170,245. Okay, so this is the number you're going to want to write down. Okay, and then after you have the this written down, you need to look for the protein product. So there you can go to the NCBI, that second link, type in Homo sapiens BRCA1, make sure it says protein here, and then search it. And you'll want to come down here. This is a protein, BRCA1 Homo sapiens. So click there. And this will give you all the information you need here. You'll need to write down the first 20. These are the single letter codes, and looks like they're in groups of 10 here. So MDLSALRVEE VNQNIV, I'm sorry, VQNVINAMQK. That's what you're going to write down. These are the first 20 amino acids. These are single letter codes. That's something you want to write or remember. And then after you BRCA1, I think I have you do insulin next. So insulin, search for that. Here we go, insulin, protein coding, and scroll down. This is on chromosome 11. It's on the P arm, so it's in near the beginning here. And you'll see here, you'll find it 2,159,799 to 2,161,341. And there's a second, like we had before, there's a second area where you can find it. But just write down that first location. That's good enough. But just gives you a sense of how many how many nucleotides long it is. We see these are all the addresses, the P arm here, section 1, see area 1, section 4, subsection 1. I think that's how that goes. And then after that, go back to the NCBI, and you can do the same thing, insulin. Homo sapiens insulin, and search that. It's going to search automatically. Come down here, insulin Homo sapiens, and scroll down. You'll see here much smaller, and write down those letters. Okay, so that's a little tutorial how you use the GeneCards website and the NCBI website. And let me get, I think I wrote down what you need to write in here. I'll do the first, the BRCA. Hope this doesn't go. There we go. I think so. The locus, chromosome 17, and so that's the chromosome. It's 43 million, 44, 295 to 43 million, 170, 245. So think about that. That's, millions, tens of millions of chromosomes long. And I did, I talked about the location. I'll come back to that, but you have the Q arm and the P arm. Q 21, subsection 31, and then you're going to write down that polypeptide product. So MDLSALRVEE, and those are single letter codes for proteins again. Normally we use a 3-letter code, right, when we use our normal protein synthesis data table or chart. But when you do an online database like this, you don't have time for all those letters, so you have to simplify it to that single one. So VQN, I don't have time for that stuff. EN, VIN, VINAMQK. So the same process you're going to do to get your insulin and your blood type. And okay, so this is what I was talking about. So here you would read this chromosome 3, region 2, band 2, subband 1. I could not remember that in that video, but to save my life. So chromosome 3, region 2, band 2, subband 1. That's how you would read that address. And if you see, they do count. It's interesting. You start the centromere and you count towards the ends. Okay, you don't start from the P band. And I mentioned that in the video. I misspoke. This is where the numbers start, and you count this way. Okay, so these are the smaller numbers of the small locations. These would be the larger kilobase pair location numbers. And you may have heard telomeres. It's kind of interesting. These gradually wear away when you get older, and these tips get smaller, and that's what researchers believe leads to a lot of cancers, those telomeres getting smaller over time. So make sure you label chromosome, something like that. Chromatid. This is the centromere. And say, for example, this is a gene, would be the locus. And might as well Well, let's see. I mean, I'm scared if I touch those buttons down there and try to erase them, I might get rid of the whole thing. Here, it's been copied. We've looked at that before lots of times, trying to impress upon you that those are the same, just one has a copy of itself. And these two would be called sister chromatids. And that's basically what you need to know. And why prokaryotes never diploid? You only need diploid to separate for things like mitosis. And since prokaryotes do not use mitosis, so mitosis, no, they do something else. If you remember, it's called binary fission. Okay, so you only need you need to have a diploid number, meaning you get a half number, so you can divide things up. Prokaryotes do not do that. They just have that single chromosome that gets copied and then separated, binary fission.